So thanks everybody for attending. This is the second seminar in our first annual LTER Network Office webinar series. And from now through June, we'll be doing these monthly webinars, and you can get the schedule on our new and improved website. So we currently here at the National Communications Office and NCEAS have 6 active synthesis working groups out of the LTER program. They're all at various stages of work, and the groups have come together primarily around the LTER program topical research topics: productivity, population dynamics, disturbance, and so forth. So I'm going to introduce our speakers for this webinar in a minute, but first I'm just going to do a little bit of housekeeping here, or a few bits of information about Zoom in case, for those of you who are not that familiar with Zoom. So you can ask questions of the speakers, and today in our webinar, our speakers are actually going to pause a couple times during their presentation to allow questions, and they'll also be handing off the webinar to each other for presenting. So you can you can use the question and answer button. It's in the meeting controls button at the bottom of your web interface there, and so you can type questions throughout the presentation, and those will pile up, and when we pause for questions, we'll be able to curate those and pass them on to the speakers, or the speakers will be able to answer. There's also a raise your hand button, and you can use that as well, but what we would prefer is that everybody mute themselves and not do any kind of verbal questions and until the pauses. Okay, the other thing is that the presentations are recorded and they'll be put up on the website afterwards, so please share that widely. So it's my pleasure today to introduce Kimberly La Pierre and Meghan Avolio and Kevin Wilcox, who will be talking about some of the early results from their working group. Kim is at the Smithsonian Environmental Research Center. She's a community ecologist and focuses on ecosystem responses to global change. And Meghan is an assistant professor at Johns Hopkins University in the Department of Earth and Planetary Sciences. And Kevin is in Fort Collins, Colorado. Love webinars. These guys we're all sitting in different places at the USDA. Kevin's also interested in global change drivers. So and the name of the topic of their working group is Global Patterns in Stream Energy and Nutrient Cycling. So with that, I will turn the floor over to our speakers. And again, remind everybody to mute your microphones. And if you want to ask questions, either type them in or wait for the breaks. And we'll get to those. Okay, thanks.
Thanks, Jen, and thank you everyone who's tuning in. So as Jen said, my name is Kim La Pierre, and I'm joined with my colleagues Meghan Avolio and Kevin Wilcox. And we're going to be telling you today about our working group that's hoping to build capacity to increase interactions between modelers and empiricists, which is often kind of a scary or daunting thing for many people, especially like I am coming from the empirical side. So hopefully we'll be able to show you that it can be done, and we're working towards actually doing these things. So Kevin, Meghan, and I all started out our careers as field ecologists, empiricists. So we go out every summer to the Cons of Prairie, LTER in Kansas, where we have ongoing long-term experiments manipulating various different global change factors to see how the ecosystem is going to respond. And so one of our experiments that we've worked in for a long time is a nutrient addition experiment, adding nitrogen and phosphorus. And we've seen in this experiment initial productivity responses to adding nutrients. We see more grass growth when you have more nutrients available. But we've also seen really drastic changes in community composition. And so you can see in this picture that we have here very clearly where the different plots are. You see squares of different species of plants, and they're all growing much more than the squares adjacent to them that do not have nutrients added. And so this is really where we got our start thinking, and the motivation behind our working group is how are these community changes that we're observing in the field impacting the productivity responses that we're seeing in this system. And we can look across a bunch of different nitrogen addition experiments from all across the United States. in many different grassland types. So we have agricultural grasslands, annual grasslands, desert, music, and montane grasslands. And as we look through time on the x-axis here, you see that there's an increase in the change in community composition through time on the y-axis. So all of these different grasslands, as you add nitrogen to them, are experiencing some sort of community change. But what we also see is that over that same time period, the response and productivity of these same grassland systems is very variable. So some of them have an increase in production, some have nonlinear responses, and the community changes that are very consistent don't translate to these functional changes in the same way. And so the goal of our working group is trying to examine what's causing the productivity change and can we link it back to a community change. So to do this, we've assembled a great team with the help of the LTER funding, as well as Meghan had a postdoctoral fellowship with SYSYNC that funded some of this work that you're going to see today. So we have many working group participants, both empiricists as well as modelers, that we're bringing together over a series of 3 different working group meetings. So just a subset of us are pictured here from our first meeting, which was bringing together the empiricists to really look at data that's been collected from the field and try and synthesize community and ecosystem responses. Our second meeting was in Fort Collins, and none of us were smart enough to take a picture, unlike when we met at NC's where they had us all take a picture. But we brought together in the second meeting both modelers and empiricists to really try and get each other on the same page, find a common vocabulary so that we could talk about incorporating community changes into Earth system models. And we have yet to have our third working group, but that will be focused mostly with the modelers. But in addition to the people that are coming together for these working groups, we also have 63 additional people who have provided us with data to build a big database of global change experiments from around the world, as well as people that have been working together with Meghan Avolio to develop an R package to incorporate metrics of community change that everybody can use at home. And she'll tell you about that a little bit later. So the key questions of our working group are to examine how plant communities change with global change drivers and what the best metrics are to capture this change. So Megan's going to talk to you a little bit about that today. And then, do these plant community shifts impact ecosystem function? And I'll tell you a little bit about that. And then finally, what are the best approaches for incorporating these shifts in the community into process-based models so that we can make global predictions about how ecosystems are going to function in the future? And Kevin's going to tell us a little bit about that. But our overall goal is to incorporate these community changes that we know are happening in the field into process-based Earth system models. And so our approach to do this began with compiling 101 different resource manipulation experiments from around the world. And this was actually funded by a previous LTER working group back in 2012. So we compiled all of these data sets. It took a long time to get them all together, but they span a wide range of ecosystems in terms of climate, as well as biotic factors like productivity and site level diversity. And we can now use all of these data sets to synthesize responses at the community and ecosystem level. And so to start off, Megan's going to talk to you a little bit about the different empirical approaches that we are using and developing to estimate these changes that we're seeing in communities.
Great. So when we first started thinking about this back in 2012, when we first got funded, we were really interested in trying to understand patterns of community change. And we wanted to go deeper than just species richness, because we could envision the situation where we have 5 species in the control plots and 5 species in treatment plots. So richness is 5 in both situations, but there's actually a turnover and every single species has a different identity. So we wanted to look at overall community composition, and we thought multivariate metrics were a great way to do this. So I want to just orient you to some multivariate metrics before we go any further. So showing here on this graph, I have some control replicate plots, and how close 2 points are to one another demonstrates how similar they are in terms of community composition. And for this, we are using, or generally our methods that we use are Bray-Curtis, similarity or dissimilarity, and that has to take into account species and their abundances. Okay, so these are all the controls, but we could also imagine that there's treatment plots shown here in black. And the next thing we can do is say, okay, how different are these treatment and control plots from one another? And so to do this, we calculate the centroids of both treatments, and we look at the distance between the centroids in this multivariate community space. So this is a measure of overall composition differences among the 2 plots. How similar are they in terms of composition? But there's another aspect of information we can get from this. We can also look about into the dispersion around the centroid mean. So this is how far is each replicate from the centroid of that treatment. And so here we have probably similar levels of dispersion between these two treatments. And so again, we were looking at both compositional change and dispersion change. And we were thinking that there's many ways that composition and dispersion can change. So composition here is overall mean change. Is there a change in the overall community, and is there a change in variance? And in the first panel, we see here that there's no change in either. So the community has not changed in response to whatever treatment there is. But in the bottom, we said there is a change in mean, there's just no change in variance. And we can have all possible pairwise combinations. So we can have both an increase and a decrease of variance without a change in mean. And we thought this was potentially important because if you have an increase in variation, every replicate could be doing something different. It's going to be very hard to predict how the community is changing. Conversely, if every replicate is doing the same thing, we have strong convergence, it's going to be easier to predict. And I just want to mention that when we're talking about increase and decrease of variance and change in mean and no change in mean, we're talking about two aspects of beta diversity. We're talking about variation beta diversity when we talk about dispersion or variance, and we're talking about turnover beta diversity when we're talking about change in means or the composition. And as a working group, we decided to really try and get away from the terms of beta diversity because they have different definitions and different ways of being defined and measured throughout all ecology. So to get away from those terms, we're really focusing on mean differences as community composition and variance as changes in dispersion or heterogeneity of replicates. Okay, so we had these 6 hypotheses that we envisioned that communities could respond to global change drivers, and we went out into the literature to find examples of these, and we were able to find an example of every situation, from benthic annelids in response to nutrient additions to mollusks underneath gas drilling wells to old fields and abandoned forests, as well as ant communities and in insect communities in response to invasive plants. And so across all these different things, all these different global change drivers, we saw exactly what we expected, to have an increase or decrease in dispersion and change or no change in composition. And so these were just 6 examples. We wanted to go out and say, okay, overall, using all the compiled data sets, what do we see? What can we find? So to do this, we're using the 101 compiled data sets that Kim mentioned earlier. And we're looking at 2 things. We're looking at trends in species richness, which is just the number of species, and we're looking at changes in multivariate community space, so changes in composition as well as changes in dispersion. And to do this, we ran a hierarchical Bayesian model looking at treatment type as well as interactions with these site level parameters that Kim mentioned. So for each line that you see here in this richness graph, I just want to orient us for a second. It's always comparing the control to that treatment. So it's always a difference. It's what is the difference from the control to that treatment. And we ran the model, and overall we find, first off, that there's no difference between the different global change factors. So we looked at things like carbon dioxide, nitrogen, water addition, water subtraction, temperature, clipping, you name it. We probably have it in our database right now. And there's no overall difference among all these different global change drivers. They're all having the same effect. And for richness, you see there's lots of variation in the light gray lines as every single different experiment, but the overall global mean of the model is that there's no change in richness. So while we see lots of variation, we see no overall trend with richness. There's no systematic response. And then, so that's richness. What for community composition? What we find for community composition is that there is a strong increase in community composition change over time. Now, again, what's so interesting is that there's no difference among the different global change drivers. Regardless of what you're adding or taking away from this plot, it's having the same response, but it is systematically increasing the community composition difference from those controls. And lastly, for dispersion, we find, again, no overall systematic picture. We find many examples of divergence when they're becoming more different, or in convergence when they're becoming more similar, but no overall pattern. So this is really great. This is exciting, right? We're seeing that these communities are changing, but how do we actually take Bray-Curtis change and incorporate those into models? Bray-Curtis change is very abstract. It's just the distance between points in this multivariate made-up community space. And so we can't take that and necessarily put that into models. And instead, we have to tease apart what exactly about the community is changing. And as I showed you in the previous slide, it's not species richness. We need more nuanced measures of community change than just richness alone. So to do that, we think theoretically that rank abundance curves are a great way to study community change. So there's 5 ways that community can change, and I first just want to lead us through this little cartoon rank abundance curve. So here we have on the x-axis is the species rank, and on the y is the species abundance, and this is at a community at time 0. And we see that the most abundant species, this purple species, has the highest abundance. So first, a community can change in its evenness. And you see that here as the line has become flatter. So now the abundance of all the species are more similar to one another within the community. There's greater evenness overall. The next we can see is a reordering of species. And in this example, now the most abundant species has become the least common species, and the rare species has become the most common species. So there's a reordering and a change of ranks within the community. We can also have a loss of species. Now we lost the light blue species, and we can also have a gain of species. And here we gained this red. And I'd like to mention that both a loss and a gain of species are associated with a change in species richness. So these are these 5 ways that communities can respond to global change drivers. And there's 2 ways to make it a little bit more confusing. There's 2 ways that you can study these changes through time, or you can study these community responses. You can study changes through time. So this is an example of control plots, and we're looking at one replicate, and we're going back to that same plot replicate each year and studying how is it changing. And a lot of ecological community data sets, this is the way you're going to analyze your data. It's only observational, like most successional data sets, and you can just go back and say, how are things changing over time? And if we're in an experiment, we can do the same things in treatment plots. We can go back to the same replicate within a treatment plot and saying, how is this changing over time? But what's really exciting about experiments is that we can also look at differences. So at a single time point, we can say, how are the treatment and the control plots different from one another? Now, these are theoretically the exact same things we're asking. How is the community different? Is there a gain or loss of species? What are the species differences? How are the ranks differences among these communities? But there are theoretically different approaches to studying them. And so changes are a change in the studying a change into plural, while differences are a change in spatial dynamics. Yeah, so we have changes over time. So how should we study all this? So I've probably overwhelmed you with all the different ways I think people should study community changes, but good news, we're working on an R package to make this really easy and accessible for the ecological community. So we're going to add an update to the existing code in R package, and we're going to introduce these new metrics to look at both changes and differences in communities. So as I said, they're the same idea. It's just either there's a difference or a change, and the only thing that is really different is that you can't have gains and losses when you're comparing comparing 2 plots at one time point. They're just species differences, and it's not clear what species was gained or lost from which community. So we have all these different metrics. To test how these metrics are related to one another, we're using an observational data set. Can we go back a slide, Kim? We're using an observational data set of 68. We're using a data set of 68 observational data sets. So for these, we can only study changes through time. And then to compare them about how you use them in an experimental context, we're going to use an example of an experiment and look at both change and difference metrics together. So first, for just the observational data set for change alone, this is a little overwhelming. I'd like you to just focus attention on the data that I'm showing in the highlighted red box. And what we're looking at is what is related to compositional change. So earlier, I showed you from all our global change experiments, we found that the most thing that was changing was community composition. But what aspect of the community is driving that composition change? Well, we see that for richness, there's everything significantly correlated. That's what the asterisk notes. And the number in this box is the R, so the strength of the correlation. So we find that while everything is correlated, both richness and evenness are very weakly correlated with changes in composition. Species gains and losses are also more strongly correlated, but the strongest correlation overall are rank changes. So it's reordering within the community that's driving most of this change in composition that we are observing. So that's using change data sets over time. How about in an experimental context? So in experimental context, as I mentioned, we could both do changes through time, or we could look at differences. So here I'm using the same experiment Kim mentioned earlier, the p-plots experiment that's located at Prairie. And we see here are the control plots in the pre-treatment year in black, and the 12th year experiment is shown in red. And you see they're occupying pretty much the exact same space in community, multidimensional community space. So there's really just no change in the community. But if we compare that to the nitrogen phosphorus addition plots, we see that there is both a change in the mean and an increase in dispersion among these. So, okay, what exactly about these plots are changing? So now we're going to use the first approach, which will look at the changes through time of the different replicates in these treatments. And so when we do that, we have the change of each replicate to itself, and then the standard error of change within that treatment. we see that for richness, there's really no big differences between the nitrogen and phosphorus addition plots and the control plots and how richness is changing over time. And there's really no big differences in how they're changing in evenness over time. It's very similar. For rank change, we see that initially there's not much difference, but then starting around 2006, we see much bigger rank changes over time in the nitrogen and phosphorus addition plots compared with the control plots. So every year there's greater reordering within this community that are experiencing these nutrient additions. And for species gains and losses, again, we don't really see much differences between the nitrogen and phosphorus plots and the control plots. So again, it really seems that when we're looking at change, what the biggest difference among them is reordering or rank changes within those communities. But that is change. We can also look at difference. So when we look at richness differences, there's really, again, not much going on. There's lots of variability from year to year. And I just want to point out that this is now a comparison. So we're comparing the control to the nitrogen plots. So what is the overall richness difference between these? Maybe there's a trend, an upward trend with this data, but again, it's very variable. And for evenness, again, it's very variable, but there's really no overall differences. For rank difference, we see the biggest change. So initially there's not much difference between them, but then over time, there's much bigger rank differences or reordering within these two communities. So if they have, the communities are similar, but whose abundances are up or down are different between these two communities. And for species differences, we see that there is an increase in species differences over time, but they're also very mild. So it's potentially that species differences are becoming more important over time. Okay, so both the changes and differences, ways of looking at community change, show that reordering is an important aspect of community change that's driving these compositional responses. And our next steps are to use all 101 experiments and to, with these, study how communities are changing and responding to the global change drivers over time. So this really shows we understand how communities are changing, and the next question is, how do we work this into models? And so I think that's it, and I'm happy to take any questions.
So just to remind everybody that if you'd like to ask a question, you can look for the Q&A tab below on the lower bar of your screen, or you can look for the raise your hand button, and we'll see that and we will unmute you because actually you all are muted by us, and you can verbally say your questions. So those are how you can ask your questions. Or type it into the chat. We'll see that as well. But make sure you address your chat to the panelists and the other attendees. That's fine. 3 ways. And Megan, I'll give it an extra minute or so since it's logistically a little bit harder for people to figure out where to ask the question, and then we'll move on.
Great.
Okay, so I see no burning questions at this point, and we can circle back. Oh, I take that back. There's one in the Q&A. Now, Megan, you should be able to see that. Can you see it? Okay.
So the question is, can you explain the results about species richness and species differences, please? Really great question. So changes in species richness are to a single plot over time. So we could say at time 1, this plot had 5 species. At time 2, it had 6 species. So the change in richness is a difference of 1 species. Then when we're looking at species differences between 2 treatments, it is what's the composition of species in plot A and what's the composition of species in plot B at time 1. And the number of species that are shared or not shared would be the species differences.
So I think one important thing to point out there is that species differences takes into account the identities of the species in the plots, whereas richness might not. So you could have 6 species in 2 different plots, but they could be 6 totally different species in each plot. So you'd have a difference of 6, but the same richness value. So the richness difference would be 0, the species difference would be 6.
And great, so the next question is the main effect of the first metric you mentioned. You include that the response for all experiments with the graph shows great variability of the results. And the overall average line is black. So what we did, we ran these multivariate, these Bayesian models, and we took the model average across all the treatments. And the first thing is we didn't have to look at differences among treatments because there were no statistical differences among the different global change drivers. So we could just take an overall model mean of how is the community changing, which was what's shown in black. So yes, there is great variability there, but that variability is not being driven systematically by anything. Oh, I think this missed this, but where are the experiments from, and how even is the richness in the experiments? Well, that's a really great question. We actually didn't say this. So all the data sets are from herbaceous plant systems, so they have to be terrestrial and it has to be plants. And you had to have manipulated a resource, so either light, water, carbon, or a nutrient. And in addition to that, you could manipulate anything else. You could manipulate temperature or herbivory. Sky's the limit. And richness varied greatly because we have some Arctic sites and salt marsh sites that have very low species richness to tallgrass prairie, which has very high species richness. And sorry, Arctic sites also has high species richness. I meant to say salt marsh only.
Great.
Great.
So I think, thanks, Megan. We can keep going here. Maybe. Yes, okay. So as Megan said, we know that these communities are changing in this big global database of experiments that we've put together, but now we want to be able to link those changes in the community to changes that we might see in ecosystem function. So people have been thinking about this for a while, that we might see direct responses to environmental drivers in terms of ecosystem functions like productivity, carbon storage, nutrient cycling. But we also might see changes in function that are mediated by biodiversity. So the environmental changes causing changes in diversity that then feed up to influence ecosystem function. And Smith, Knapp, and Collins back in 2009 put together a really nice framework to try and tease apart these different community drivers. So they call it the hierarchical response framework. And here we're looking on the x-axis through time and on the y-axis at the ecosystem response, whatever that response might be. And initially, you might expect that you only see individual level physiological responses. So individual species might grow more or less because of whatever environmental change driver they're experiencing. So you might imagine if you water a plant more, it's going to grow more. That would be an individual level physiological response. But they posit that those responses must be limited by the physiology of these individuals, so it can only be so big. So through time, as you start to see community changes, then you might start to see a stepwise increase in that ecosystem response. So first, they think you'll initially start to see species reordering within the community. So the existing species in the community could be changing. That would probably happen faster than species immigration or species loss. And because of the different traits of those new species coming in, you could see a greater ecosystem response. Further, you could see even further in time, species immigration or additionally species loss that could again be causing bigger ecosystem responses than you could see from individual responses alone. And importantly, they point out that these responses could be stepped in nature, they could be linear, kind of all blending together, or you could skip stages. You might only stick in the individual physiological response stage for a really long time. You might skip reordering and jump straight into immigration. And all of these changes in the pattern might result from different types of communities that you're working with. And so a working group led by Melinda Smith and Alan Knapp back in the 2015 tried to get at these ecosystem responses to see, do we see these various different patterns? So we looked to see across a bunch of different LTER data sets, terrestrial data sets spanning forests to herbaceous systems, looking at global change experiments. Did we see ecosystem responses where they were transient? So you might expect to see an initial response that goes away eventually. You might expect to see stepped responses, so potentially a lag and then a bump up in your ecosystem response. You might expect to see a continuous directional response, potentially with some steps in there, or you might see no response at all. And in our synthesis, we looked here at the difference between treatment and control plots and productivity across a bunch of different LTER experiments, and we did see examples of all of these different response types. So in the Virginia Coastal Reserve salt marsh system, they were looking at changes in sea level by pumping water out or in. and they saw no response in productivity to that treatment. In the Sevietta grassland, they were adding nitrogen, and this is a desert grassland system, so they saw transient responses where in years where there was high water availability, they saw a big ecosystem response to nitrogen additions, but those responses went away when you didn't have high water. In the McMurdo Dry Valley, they were adding water and temperature, increasing temperature, and they saw big ecosystem responses after a significant lag in response to temperature alone or temperature and water addition together. And then finally in the Arctic tundra, with nitrogen and phosphorus additions, they saw, again with a little bit of a lag, a pretty linear increase in productivity in response to nutrient additions. So we definitely see evidence for lots of different shapes of ecosystem response curves. And this seems to provide some evidence that maybe there's something going on with the communities in these systems that are causing the shapes of these responses to be so different. And so our working group is really trying to link these community changes together to examine how they're affecting the ecosystem response. And so we've come up with a conceptual framework to try and tease apart the community responses from the physiological responses, where we can imagine at the bottom level, we have some sort of global change factor that could either influence the ecosystem response through a physiological response on the left, or through some of the community responses that Meghan mentioned in the previous section. So we could see changes in species evenness, reordering of the species within the community, or gains or losses of species through time. And overall, both of these two pathways could feed up to influence the ecosystem response. And so today we're focusing on primary production as our main ecosystem response across these herbaceous global change experiment data sets that we have collected. And so we've turned our conceptual model into a structural equation model to try and test the different strengths of these pathways. So as Meghan mentioned, we have lots of different types of experimental manipulations. The ones we're focusing on for this analysis are nitrogen additions, phosphorus additions, potassium additions, water either increases or decreases, so irrigation or drought experiments, atmospheric CO2 increases, and then we have another category that encompasses a whole range of different factors. And all of these treatments could individually impact directly productivity responses, so that's the big curving arrow on the left. And if they did that directly, we would expect that we would be seeing some sort of physiological response to these treatments. In contrast, they could also feed up through our community change metrics. So we could see changes in the rank difference, so that's reordering within the community, evenness difference, species difference, and richness difference. So again, here we're comparing treatment to control plots, all in space within the same time point. All of those community metrics could feed up to influence our Brake-Curtis dissimilarities, or our multivariate metric of community change, which could then impact ecosystem function differences or productivity differences in our case. And so what we have is, for all of our data sets that have both productivity and community data, we've put them all into one big model, and we're looking at 10 different models through time, from pre-treatment all the way up to the final or a median year of our data set, so that we still have enough data to be able to run these models. So today I'm just going to show you what happens at the first year and what happens at the 9th year of data as an example, but we're running these models through time. So for all of these models, we include all of the paths in every model, but here I'm only showing you the significant paths just for simplicity. And in the first year of all of these experiments, what we really see is direct physiological responses of productivity to our global change drivers. And really they're primarily responding to nitrogen additions and these other treatments. So again, these responses in the first year are really driven just by the physiology of the plant's individual responses. And we can compare that to the 9th year of the data, where we start to see some really interesting patterns. So we still do see physiological responses of the plants, and they're about the same order of magnitude as we saw in the first year. So all of the different treatments did have direct effects on productivity at this point, but I'm only showing the two biggest, which are the nitrogen additions and again, the other treatments, both having about the same effect as they had in the first year of the experiments. But what we also see is that all of these treatments are really having a big impact on our community metrics. These community metrics are then feeding up to influence the compositional difference in multivariate space. And primarily what we're seeing is a big change as Megan showed in her previous slides, in the rank difference. So this is reordering of the species in the community, as well as the species differences. So how many species are shared between the control and the treatment plots in each experiment. And these changes in the community are really feeding up to influence the productivity differences that we're seeing within these different plots. So at the later time points of these experiments, we're not seeing so much the physiological response, but rather a community change that's driving the big productivity differences that we're seeing. And so this is really providing evidence that this hierarchical response framework may be, in fact, representative of what's going on in these ecosystems that we're studying. We initially see these physiological responses, and through time we start to see species reordering, changes in species either through immigration or species loss. And potentially if we looked at these experiments after 20 years, which we could do for a subset of them thanks to the LTER network, we might start to see even more or higher rates of species immigration and loss. So through time, we'll be able to look at these changes and show that the stepped nature of the ecosystem response is really visible in many of the ecosystems that we're working in. And so overall, what we're seeing is we have community changes. Those community changes are leading to ecosystem responses. And now we want to link those together into these big Earth system models to try and make big predictions about what's going on in the world. So we'll pause again for questions if anyone has any before we move into the modeling section.
And to stay on track time-wise, I think we have a couple minutes for questions.
Great, thanks. And just to remind you, either use the raise your hand button and we will unmute you, or type your question in the Q&A box. Okay, Kevin, there's Kim, there's a question.
All right, so Nate asks, in the SEM, the physiological response is a direct effect of nitrogen or phosphorus on AMPP, and have we considered putting in a latent variable for physiology in the absence of direct measures? So we definitely have considered this, and we've tried it out. We don't quite have enough data to have all of these different factors all flow through a latent variable as well. So we took it out, but we're definitely considering how to improve this analysis. I think another thing to think about is we have the physiological response of individuals and the population response of species. So there could be changes in density as well as changes in growth of one individual, and teasing those apart in these models is something that we're interested in doing, and we're not quite sure if we have the data to do that either. And then one other question is, did we test ecosystem function effects on community composition? So going the other way, presumably. So in previous analyses, we did do that, the ones that Megan was showing. We looked to see, is there an effect of site level productivity on changes in composition? And we don't see that effect. We do see a little bit of effect of site level diversity. So communities that have higher site level gamma diversity see greater changes in response to ecosystem functions, but we don't see a correlation with site level productivity. But we have not reversed this structural equation model to point the arrows in the other direction and see if the productivity response then affects the community, which is very possible in terms of thinking about light availability, changes in soil moisture, things like that. So that's a great idea. All right, so Kevin, you want to take it away?
Yeah, so the next step was to try to incorporate some of these community shifts and their impacts on ecosystem functioning into process-based models. And so there are a lot of theoretical models out there that have been used in the past to predict how communities might change with just through time and with changing environmental variables. A lot of time, things like R-star theory, a lot of times these theoretical models, though, lack a robust link from these community shifts to ecosystem functioning. And this is where process-based models come into play. I should say biodiversity ecosystem function theory does provide that link, but as Meghan and Kim showed above, a lot of these changes that we're seeing aren't just richness, it's actually changes in the identity and the dominance of different species. And so process-based models can help to try to scale from this physiological level to the ecosystem level through these community changes. And this is tricky. A lot of times with empirical data, we try to scale from these smaller hierarchical levels to landscape levels, and it can be challenging. So process-based models are a great tool to do that for a couple different reasons. We want to make better projections of carbon cycling dynamics, atmospheric feedbacks, albedo, things that these big Earth system models have kind of been designed to focus on. But also, we can use these tools to test hypotheses about how communities will change through time and what impacts that might have on the functioning of ecosystems. And so there are a whole, there's a wide variety of different models out there that vary in how complex they are, what kind of parameters they use to represent natural or physical processes, the structures that they use, and theory and data typically drive these different models, and they can be used for different things. If you care about hydrology, you may use a different model than if you just care about phenology. But theory drives these models. Then collectively, we can start to get ideas about how ecosystems might respond or exist in the future, and that can guide management decisions. And then an important part of this is when you run these models to make projections, you can identify limitations in the model structure, the parameterization. No model is perfect. They're just, the ecosystems are too complex for that. And so that process actually can guide future data collection and experimentation efforts, which goes back into developed theory and then feeds into a new iteration of models. And so I wanted to just walk through a way to go about developing models to incorporate these more fine-scale vegetation shifts. And one of the first questions is this even important to incorporate the process-based models? From our second working group, we really found a difference in the way that community ecologists and process-based modelers think about ecosystem processes. The modeling side thinks very heavily in terms of physiology, because that's how the models are programmed. But a lot of community ecologists think about abundances of populations and communities, or abundances of species within communities. And so, one question is, do we even need to think about incorporating a more community ecology approach into these models? And so to do that, we ran model simulations and then compared those simulations with global change diversity global change experiments. And then the second, there's multiple ways that models can incorporate vegetation dynamics into their projections. And so thinking about how these different vegetation change approaches inform different scenarios. For example, you might want to use traits to model community change through time if you have like a steady shift in resources. But with disturbances, maybe you need to model individual organisms within the community that are competing for light and things like that. And so the next step is to then compare these different vegetation dynamics in GCD. You can advance it, Kim. Examine these different simulations of vegetation change and then ask the question, what types of data do we need to implement these different types of vegetation change? And finally, it's important to have discussions about what data are needed, what model updates are needed, what's available, and what's honestly realistic to go out and collect in the field. And so the specific goals of this second meeting were, one, what different types of process-based models are out there? Two, do we even need to improve these process-based models to incorporate more fine-scale vegetation dynamics? Because current models, they do plant functional type changes in dynamic global vegetation models. what processes specifically do we need to improve, and then what data are needed to inform these processes. And so this took a lot of talking between groups, and just the terminology itself was difficult. The differences between terminology was difficult to overcome. But after a good day and a half, I think we got on the same page, and then we really started making progress, which is really exciting. Yeah, so this is a list of the process-based models that we're including in this exercise. These models range from more or less simple, relatively simple ecosystem models that don't actually represent changes in plant functional types or communities, to demographic models that that represent populations and how these things interact. This has about 10 parameters that represent species, to global ecosystem models that represent either plant functional types or plant traits. These use about 20 parameters to represent species, to these very complex global demographic models that incorporate demographic processes into these global ecosystem models, and these have about 30 to 40 species or parameters each that represent different species. And this is a work in progress. It's been a big challenge, but also very rewarding. So right now I'm going to show results from an ecosystem model and a global demographic model. And so the next step was to compare model simulations to actual global change experiments. And so the experiment we selected for this was a long-term irrigation plot or experiment at LTER, where irrigation, these plots were irrigated for over 20 years, and after 9 years, you had a clear shift in the dominance from Andropogon gerardii, a C4 grass, to a different C4 grass, Canna imbricata. And we saw a drastic change in ANPP after the community shift. And so I'm showing the observations as bars on that bottom graph, and some preliminary model simulations in the green dots in those. So you see before the community shift, the model does a pretty good job about matching up with observations, and afterwards it does a little bit poorer. So the next step was to use some of these models and do this in a more robust way with models that actually were supposed to incorporate vegetation change. And so we collected a whole slew of different lines of information for these different species, ranging from physiology to morphology, how the different species allocate biomass, leaf economics traits, reproduction and survival. And this is a combination. These data came from a combination of the TRI network, but also individual measurements taken at this site. Okay, and so then basically trained the models with control data, and ran the models using hourly weather data. And then once the models were calibrated, we then applied the treatment that actually the system underwent into the model looked how it did about matching observations. And so, on the x-axis here, we see observed above ground net primary productivity, and on the y-axis we have the simulated from the models ANPP, and that one-to-one line there. If the model is perfect, it matches that one-to-one line really nicely. And so we're getting pretty close in the ambient community on the left before that plant community change. These two models did a fairly decent job matching observations, but then after the community shift, we see, first of all, the ecosystem model that doesn't incorporate any vegetation change. Really, it struggled to capture both the variability from year to year, as well as it underpredicted, production. The ecosystem demography model, you see a little bit more spread on that y-axis, which is a good thing, but we're still underpredicting, that the observed ANPP. And so, so these models, these process-based models, they scale from plant physiology to ecosystem processes through parameters that represent this physiology. And community ecology typically uses about the abundances of species and biodiversity and plots to scale from the community into ecosystem processes. And one challenge that we have, and not a lot of research out there, at least to our knowledge, focuses on how plant physiology feeds into community dynamics. And once we get that through these trait-based models and these individual-based models, we can then start to scale from the physiological level through community dynamics into the ecosystem processes and have better representation of long-term trends in ecosystems. Okay, and so, and so just to go quickly into this, the last thing we wanted to do was to talk about the goals of what we wanted to incorporate into models. So we want better water dynamics in the models, better nitrogen dynamics, better representation of herbaceous system. A lot of these models typically represent trees, and you know, in these models, you know, grasses are either represented as little trees or like a green slime. So we want better representation of that, and overall better incorporation of these community processes at finer scales. So we pitched that, and the people that worked with process-based models said, okay, here are the data we need. And so to incorporate better water dynamics, we need continuous soil moisture in a lot of experiments. We need ACI curves, detailed physiology. We need rooting profiles. I'm not going to go into all of these, but it's a pretty long list. And so hopefully, these things we can collect in the future to go forward in improving these models. And then what the modeling community, I think, needs to do is to try to incorporate these community processes, both due to better water dynamics and better nitrogen dynamics, but also incorporate competition among species and among populations. And so the next steps where we're going forward is we're going to go out this summer. We're going to go out and actually collect these data so that we're not hypocritical and say people should do things and not actually do it. We're going to go out and do these things. We're going to, keep track of how long it takes and how realistic these things are. And then also we're working on a paper that we want to provide a roadmap for field ecologists and process-based models to identify, which types of models are important for which purposes, what processes need to be refined in these models, what data do we need, and then paths forward. Exciting things like using these coordinated experimental networks and observational networks to then go out and we can coordinate data collection efforts that will be useful in these process-based models. And that's exciting. And then our third working group, we're actually going to try and do some of this. We're going to improve vegetation dynamics in these models. Here we're showing a nice photo with Nate there about how we might represent grasses, bunch grasses, not as slime or little trees. So and so with that, I think any questions that we want to, or from any of the previous sections or this section, will be welcome.
Thanks, Kevin and Megan and Kim. This is Jen again. I just want to ask the participants before we get into the questions, and you've got a couple queued up there, Kevin, in the Q&A box. If you mind, could you go to participants? We've got a small poll. It will take no more than literally 3 seconds of your time. And you go down to the polling, click on the polling, or it should have popped up by now. We're very interested in understanding how you found out about the webinars and how you viewed them. So while we're answering questions, if all participants could do us the favor and take this very short poll, that would be great. Thanks, Kevin. So we've got a couple questions queued up already.
Make sure I'm unmuted. Oh, that's so many windows have popped up. All right. So the first one was about agent-based models, and that's a great question. And a lot of those dynamics are incorporated into individual-based models. And this is where CLM Fates is going, and then the LPJ Guess models are really starting to incorporate more individual-based components. The challenge is, one challenge is accurately representing coexistence in these models. And so really it's a matter of getting more information about how these different species, how they deal with differences in nutrient and resource limitation, and how they compete with each other. One ran into was that whenever we would have an individual that was access resources faster, it would drain resources down to levels where nothing could survive, or the other species that was more conservative could survive, and so it would win out. But in the real world, we know with our star theory that maybe more conservative organisms might be able to exist at lower resource levels, and that's how they survive, and that's how they coexist. So these things are all things that we need to address, and the individual-based models are certainly a way to go. And then an example of a community process that a model could include, I guess one is competition, things for competition for light, and how these different species compete for light and also nutrients. And so basically modeling that change in dominance or reordering in the community as resources change is what we mean by modeling community processes.
Okay. Well, if there's no further questions, we'd just like to thank our speakers for such a fantastic webinar. Virtually, people are out clapping in their offices right now for you guys. I know we are here at the NCO, at NCEAS. And I'd like to thank the attendees and for asking great questions. And I think, again, go to our website for the webinar schedule. We'll meet same place next month. And unless there's any last minute questions here, I'd just like to thank you, and we'll sign off.
Thanks.
Thanks, you guys.